I'm Daniel Levine, and this is the Bio Report. Gene Therapeutics platform technology Magellan leverages AI, structural biology, and physics-based models to identify novel binding sites on otherwise undruggable proteins implicated in diseases. The company's experimental Parkinson's disease therapy has the potential to slow or stop progression of the neurodegenerative condition by stabilizing a lysosomal enzyme implicated in the disease. We spoke to Gene Mack, CEO of Gain, about the company's platform technology, how its experimental therapy for Parkinson's disease works, and what other conditions might be good candidates for its platform technology to target. Gene, thanks for joining us.
Thank you, Danny. Thanks for having us, and thanks to the folks at BioReport.
We're going to talk about Parkinson's disease, Gain Therapeutics, and its AI platform for drug discovery. Let's start with Parkinson's disease, though. For listeners not familiar with the condition, can you explain what it is?
So Parkinson's disease is the second most prevalent neurodegenerative disease next to Alzheimer's. And it is a progressively worsening neurodegenerative disease whereby motor function and cognitive function decline very rapidly in the early stages of life.
And how does it manifest itself and progress?
So the manifestation of Parkinson's disease can carry out through a couple of different types of mechanisms, not all of which are clear to us. One of the routes Parkinson's disease develops and progresses is through a genetic mutation in a gene called the GBA gene. This gene is responsible for transcribing a particular enzyme called glucocerebrosidase, GCase for short. And that is the target that that's the target for our lead compound, GT02287. But how does Parkinson's disease develop and progress? In the phenotype of Parkinson's disease, let's take a step back for a minute. Parkinson's disease, our understanding of it is still evolving over time, such that clinicians and researchers are starting to appreciate the fact that there are many different phenotypes of Parkinson's disease. The outward appearance of it can be different from patient to patient. This has clinicians and researchers wondering, are there differences in the disease mechanisms among these different phenotypes, in which case several different factors are at play among the entire Parkinson's disease patient population. And it's incumbent on medicine and science to start to understand maybe there are subgroups of Parkinson's disease that need to be treated differently than others. So when you ask, well, how does Parkinson's disease start and evolve? Some of that answer can be very complicated, but on its surface, what happens in the cell is its regular ability to maintain and housekeep itself breaks down. In the case of GBA1 Parkinson's disease, it is the GCase enzyme that is responsible for the housekeeping and maintenance of different cellular functions that starts to fall apart on patients. Over time, toxic substrates that would be otherwise cleaned out from the cell build up. And those toxic substrates, as you can imagine, become more and more burdensome, and the neurons that the neurons themselves start to die. And there you have the main culprit in terms of the onset and progression of Parkinson's disease symptoms.
Well, what's the link between Parkinson's and certain forms of Parkinson's and the rare lysosomal storage disorder, Gaucher's disease, and is this a lysosomal storage disorder?
It's an interesting question and not one that can be answered simply. There's overlapping mechanisms here. In the case of GBA1, you're correct. Gaucher's disease is very, very close to Parkinson's. In fact, patients that have Gaucher's are at a very, very high risk and prevalence of developing Parkinson's disease later on. Both of which have the GBA1 gene as sort of the culprit again. In the case of Gaucher's disease, there's almost an entire loss of function in the GBA1 gene and its ability to transcribe GCase enzyme. In the case of Parkinson's disease, that mutation is not as severe in most cases. So if you imagine you have two copies of a gene for each, you have two copies of each gene. And in the case of Gaucher's, there's pretty much no functionality in either copy. In the case of Parkinson's disease, you may have some functional GBA1 genetic function. You may have some GBA1 genetic function, which leads to some functional GCase enzyme, but in most, and not as you're separated from Gaucher's in that regard. But if you are heterozygous for the GBA1 mutation, your chances of developing Parkinson's disease can be as much as fivefold higher. In Gaucher's, your GBA1 gene is completely knocked down, and the problems evolve much earlier in life and more broadly.
How do people generally come to be diagnosed?
It's driven mostly by symptoms. Patients notice the tremor, the lack of muscle control, and they can also start to notice cognitive changes. Now, one of the reasons why, this is a bit of a callback to what I said about the heterogeneity of Parkinson's disease, there are some cases where the disease presents differently. We typically think of Parkinson's disease as a shaky disorder, something where the patient can't maintain motor control. But there are other facets to this, particularly in the cognitive aspect of it. So some patients who may evolve their cognitive symptoms faster may wait longer or think it's something else. But in general, Parkinson's disease is diagnosed based on its symptomology.
And given that, would you use some kind of a confirmatory test, a genetic test, to determine which patients are correct for your therapy?
That's a really great question. In this day and age, we know a lot more about genetics than we do about how to tackle the issues that we learn from genetic mapping. And in the case of Parkinson's disease, and really any other sort of genetically predisposed disease where there's no disease modifying agents available, it's a tricky aspect. And the Parkinson's disease community is struggling with genetic counseling. What do we tell patients when we die? There's an ongoing effort to genotype as many Parkinson's disease patients as possible to learn more about things that we're struggling with now. And in the case of GBA1 Parkinson's disease right now, knowing that you may carry the mutation, there's not much we can do there's not much we can do to intervene at this point. So it raises a question of what's the benefit of knowing this. Now, obviously, we want to know as much about our health profile as we possibly can, and what the options that are available to us. But in some cases, that's not always great. It's not always an optimal. It's not always ideal. What we think will evolve if our drug GT02287 is successful in slowing or stopping the symptoms of Parkinson's disease because of its underlying mechanism and disease modifying properties, we think that will probably lead to a genetic test for GBA1 mutation that a patient who's experiencing Parkinson's-like symptoms can get, and then understand their profile a bit more, maybe make treatment decisions sooner in the pathology of their disease. If we are successful in treating early stages of Parkinson's, then it may make sense to do some genetic testing just to see if you may be predisposed, and you might start the drug earlier in your life than you normally would, just to avoid the risk. But we are far away from being able to demonstrate that through clinical trials right now. We're picking out patients based on having an established diagnosis of Parkinson's disease with or without the GBA1 mutation, because we think we can also help idiopathic Parkinson's disease patients along with GBA1 mutated patients, although they're the low-hanging fruit. We think demonstrating its effectiveness in patients already with established disease, we can then learn more about the drug as we continue to treat patients and move it earlier in the treatment paradigm, and eventually work towards maybe genetic testing patients who think they might be predisposed to Parkinson's disease and see if they have a GBA1 mutation and make some choices based on that.
And what's the prognosis today for someone with the condition, and what treatment options exist?
So there's great examples of this. The most visible one to everyone is Michael J. Fox, who was diagnosed very, very early in life and has lived with Parkinson's disease for over 20 years now. The prognosis, one of the things that is true for a lot of Parkinson's disease patients is that they don't die of the disease, they die with the disease. So you are looking at a very, very long period of time in life where your quality of life is impacted by this disease. So that's why it almost makes it, in some respects, the devastation is, not quite as terminal as some other diseases that are out there, but the overall quality of life as the symptoms progress gets far worse and worse and worse. So if we can eliminate that and provide 20 years of symptom-free, in a best case scenario, symptom-free lifestyle, then we're doing the best we can for patients. And that's really the conundrum here. Is Parkinson's disease it's a long disease. It's a long burden that patients live with later in their lives when they should be enjoying things more.
You referenced your lead experimental therapy. This is GT-2287. What is it, and how does it work?
So GT-02287 is what we call an allosteric modulator, and we'll cut through the science here a little bit. GT-02287 is meant to target the GCase enzyme as it's transcribed, and what it does specifically is when it binds to GCase, it corrects the structural integrity of the GCase enzyme. If everybody can remember from their high school biology courses, enzymes work with that lock and key model that we learned early on, where its shape, its conformational shape, is really what's responsible for its function. If the enzyme comes out transcribed and is misfolded, that enzyme isn't going to work, and it's going to cause damage because the cell doesn't know what to do with it. It doesn't necessarily, may or may not have a method of housekeeping that particular malfunctioning protein. So what GT-02287 does is early in transcription, it attaches to GCase. It improves the structural integrity of GCase, so it can traffic throughout the cell to perform all of the functions it's meant to do. And we are learning that GCase has broad function within the cell. So not only does it have to traffic through a compartment called the endoplasmic reticulum, it has to get to another compartment called the lysosome, which you referenced before, because clearly that's where the toxic substrates build up the most. And it also has to get over to the mitochondria, where it supports mitochondria complex 1 and healthy electron transport 2. So you see there's 3 compartments there that need to be, that GCase plays a role in maintaining and housekeeping. And if any one of those 3 fails, you're not going to get broad neuroprotection. So what GT02287 does uniquely is traffics. We call it a chaperone because of this. It traffics GCase, practically holding its hand as it performs all of its functions in the cell, starting at the endoplasmic reticulum, getting to the lysosome where it clears out the toxic substrates that we talked about, and then also supports healthy electron transport at the mitochondria. Again, any one of those three things starts to fail in those cellular compartments, and you're not going to have neuroprotection.
This is a structurally targeted allosteric regulator, or STAR, that was discovered through your Magellan platform. Can you explain more broadly what STARs are and their significance? What might they enable that might otherwise not be possible?
So what STARS allows us to do in the later version of a Magellan is, through machine learning, help us understand the binding and unbinding kinetics of potential molecules that can target certain proteins and enzymes of interest that are implicated in disease pathology. So what we've been able to do with STARS is introduce some machine learning, and we're still refining these things. It takes a lot of code. It takes a lot of examples it takes a lot of examples of correct binding and unbinding data that needs to be filtered in and programmed into the machine learning component before it's able to accomplish this on its own. And we're doing that refinement now as we speak. But it is capable of it's just not as we're working to make it a little bit more efficient, a little bit faster. But what STARS did for us with respect to GT02287 was it identified these certain binding characteristics that made GT02287 stand out from other molecules and other similar molecules, and that's why we were able to select it as a lead.
And talk about the Magellan platform. How does it work? How do you go from a target to a lead candidate with this?
So imagine a target protein, and what you need to be able to do with that target protein is bind to it in such a way that will either activate it or protect it from degradation. And through a series of computational permutations that measure other fragments of unknown molecules, it literally examines a protein, and then through the machine learning starts to look at the different binding kinetics between, if you were to stick a certain side chain or a fragment of another molecule into this part of the protein, will it stick? Will it change its conformational shape? And through millions and millions of iterations of this, which the computer can do very quickly, it starts to actually build out the framework of the molecule that you then try to test to see if it's got stability, if it can actually be formulated in a stable form that can become a drug. Now, I am trivializing a lot of what is done in order to get to that result, but at its basic purest form, what we're looking to do is have the machine sort of build us a framework of a molecule that we can fine tune in the lab based on the kinetic properties that the computer has determined are the most optimal in order to get binding, get structural integrity, or get activation.
My sense is that this isn't a generative platform, but instead allows you to screen a virtual library of 50 million compounds against targets you identify. How does virtual screening compare in cost, speed, accuracy relative to traditional high throughput screening?
It's a fraction of the cost. It's a fraction of the cost. It's a fraction of the time. But what it does do in some cases is we're still struggling with narrowing down the possibilities. So the compound, the risk you run is, unless the learning data is correct and is correctly input into the machine, it will not learn correctly, and it can't then predict something correctly. So before an AI platform can really be sort of launched on its own, it's got to show over and over again that it is capable of constructing something that is actually viable and can be stable and can result in a stable molecule. But in terms of the overall, high throughput screening takes known structures of compounds and libraries that are already in the public domain, or not necessarily in the public domain, but are already in many cases existed, and it basically has made sifting through those molecules more efficient. What AI has done is basically increased the potential screening ability, but also increased the target ability. So you've got this, you sort of have the world on both sides is getting bigger and bigger, and the challenge there is to narrow down all those possibilities into something that can be again formulated in a stable construct.
Is there a point of diminishing return on the size of the library? Is more better?
I imagine so. We're in the trillions now. It's gotten to a point where some of the numbers are they're almost hard to contemplate. But there's enough known about structures, pharmaceutical structures, and certain backbones and types of molecules that you probably can avoid a lot of that risk. But we also don't know what the potential is until we start to explore some of these avenues, which may run down to a path to nowhere. Again, it's all fed back into the platform as a learning experience.
And at what point do you do wet lab experiments, and do those feed back in any way into the AI system?
So one of the reasons why we've kind of, we've come up with a pipeline of promising candidates that need, more lead optimization and more work to be done in order to get ready for the clinic. But we're at the point now where Magellan has given us way more than we're able to manage in terms of what to work with in the clinic, right? So, I think at the heart of your question is, is it enough rope to hang yourself? And it can be if you're not focused on actually taking some of these molecules into the clinic and understanding, how far they will get so that your platform can really be validated. And I think you can get wrapped up in AI, and you can get wrapped up in trying to continue to refine it. At some point, you probably have to just put it to work and force it kind of to narrow down. And that's why you're still going to need some human choices here, right? So there are things that AI there's ways to. I guess the best way to put this is it's an iterative process. We allow the machines to run for a bit, see what it spits out. We put that into the wet lab, right? And then we understand more about the molecules and how they're going to exist in real environment, right? We can feed that back into Magellan, right? And then improve its learning. So the wet lab is probably, at the point where you have a stable, when you can construct a stable molecule that will be able to sit on a shelf long enough to be a clinical candidate, because that can become an issue with some theoretically designed molecules where the stability just isn't there, and that's one of the things probably that AI needs improvement on as well.
And what makes a good candidate for a star therapy, and how broadly applicable might these molecules be?
Formulation is key. The molecules, structural. If we get something that spits out, if we get a molecule spit out from the AI platform, we have our chemists, our medicinal chemists look at it, and in some cases they may say, well, that's just not doable because that compound, we can make it, but it'll fall apart as soon as we put it in a jar, put it on a shelf. Or or it'll come out with something where you can tell that the binding integrity and all of the molecule is very, very rich, and that it's going to not degrade into several different types of forms, some of which could be toxic. And then, so that's really where the medicinal chemistry piece comes in. Is this a likely, how realistic is it to formulate this? Because you can design molecules, right? But it doesn't necessarily take into account their structural stability, their stability.
What's known about 2287 from the studies you've done to date?
Great question. So what we understand about GT02287 after our first human volunteer study, so these are not Parkinson's patients, these are healthy volunteers. What we were able to learn from that study is that GT02287 is safe and well tolerated. The most significant adverse effects related to GT02287 is some transient nausea. It is characterized as mild nausea, and it is transient in that it usually goes away after about 3 hours or so in most patients. And we also notice that there's a little bit of a food effect, so patients who are fed versus ones that are fasted react a little bit better to it. But we were able to demonstrate overall it has a very favorable tolerability profile, and we were also able to demonstrate that it gets into the cerebrospinal fluid of humans. Now, this is important because we need GT02287 to penetrate the brain. And as you may or may not know, the blood-brain barrier is the most, it's pretty much the most impervious barrier there is in the body. And for the most part, lots of drugs the brain does not like foreign material and does everything it can to kick it out. So the fact that we were able to demonstrate that GT02287 indeed distributes evenly in the body and in the brain, and we were able to note that by measuring it in the cerebrospinal fluid, which bathes the brain. And if we can detect concentrations there, we know we're getting to the brain. So we were able to tick that box. I think the most important takeaway from our early studies, our early human studies of GT02287, in looking ahead, is the fact that we actually have target activation. The one important takeaway from our studies has been we were able to increase GCase activity among healthy volunteers. This is a very, very important observation because prior to running this study, understanding that these are healthy volunteers with homeostasis, all of us who are healthy have good homeostasis in our cells. If something is out of balance, there are compensatory mechanisms that work quickly to bring it back into balance. We were not sure we'd actually see. The goal of the study was just to measure if we were getting to the brain and the tolerability. We looked at GCase activation in healthy volunteers, not expecting to see anything because healthy volunteers should not have any dysfunction in GCase. So if we were even able to elevate it artificially, very, very quickly, the body's homeostatic mechanisms would bring that into balance quickly. We were very surprised to see activation among the healthy volunteers, and significant activation too, by 53%. That's not dangerous or anything like that because eventually those patients will go back to homeostasis. But it was interesting to note that we saw GCase activation. We know we're activating the target GT02287 is focused on. That is a very, very key observation, giving us confidence that the rest of the disease cascade will fall in line. What I mean by that is as we are activating GCase, we can hypothesize that we are likely also protecting its maintenance and the endoplasmic reticulum and the mitochondrial health.
And what's the development path forward?
So the development path forward is a phase 1b proof of concept study in Parkinson's patients, which we are initiating over the next couple of weeks in Australia. And here we'll learn, again, a little bit more about the tolerability and safety profile. The healthy volunteers with mild nausea, that suggests great tolerability and very, very favorable safety margins. But we actually have to take that carefully into patients as well. Parkinson's patients are going to be on other medications, and they're also a little bit more fragile, a little more vulnerable We want to make sure that that grade 2 nausea doesn't turn into something worse in Parkinson's patients. So we want to take a small number of them and test them over a 3-month period. In the healthy volunteer studies, we only dose for 14 days. In this case, we're going to dose patients for 3 months and take a look again at GCase activation and see if there's any disaggregation of the toxic substrates that should follow from improved GCase activity. So the next step of that would be, well, okay, you're activating GCase. Are you demonstrating that the activated GCase is making a dent in some of the accumulated toxic substrate that Parkinson's patients have developed over time? The later potentially clinically observable improvements that would lead to will take longer probably than 3 months, and that will be something that we start to think about in phase 2.
The company went public in 2021 at $11 a share. It's been a tough biotech market since then. Gain most recently raised $11 million through a public offering in June at $1.35 a share. Has being public made it easier for Gain to access capital?
That's an interesting question. I think it has overall. In biotech generally, capital has been very difficult to come by in the post-COVID, sort of in this post-COVID time frame. In probably late 1919, 2020, and 2021, a lot of money poured into the sector, both public and private, and I think we're still seeing some of the cooling off of that. Based on just in some investor fatigue over the last couple of years and other opportunities in the markets. And for us, it's just meant that access to capital has been a little bit more stingy, but we have been successful in raising the cash that we need. We're going to deliver our next data milestone, which is this phase 1b study. We'll have some analysis of that data in the first half of next year. We don't need to raise any capital before we're able to do that.
So how far will existing capital take you, and what is the plan for raising money from here?
Right. So at the end of the third quarter, we had $12 million on the balance sheet that is sufficient to finance operations through the first half of next year. And we'll have our next data milestone before that. Now, our approach to raising capital is opportunistic. If the opportunity presents itself like it did back last June, we may or may. That was a difficult financing. I don't think we duplicate that particular type of financing again. If we have an opportunity with an interested investor who is collaborative with us, would we seek to raise money in that regard? Yeah, very likely, maybe. But we are opportunistic with respect to raising capital. We have structured our business milestones and business objectives to coincide with when we would actually need capital. So, the market likes data. You need to deliver data. If you do that, the market is very, very loyal to you and provides you the resources to continue on. So our next sort of homework assignment is to successfully complete this phase 1b data, demonstrate more of the case for GT02287 beyond GCase activation, showing actual disaggregation of some toxic substrates, and demonstrating to the world that cascade is playing out as we saw in our preclinical models, and it is translating over to human beings. So we'll deliver that data in the first half, as I said. In the same time, we're talking to potential partners on an ongoing basis. We talk to them. We've talked to them recently on the new data that we've had. In some cases, they're mulling that over. We talk to them about other aspects of the drug on a regular basis. And our view there is we're going to continue to engage with potential strategic partners. Any deal that we do has to look as good 5 years from now as it does today. And what I mean by that is not only do we have GT02287, but we have a portfolio of backup compounds of GT02287. And some of these backup compounds, in some cases, are showing more potency than GT02287. We don't know what that means just yet, but we need to develop those drugs to see if maybe down the line we can alleviate, for example, the amount of drug that we have to give the patients with GT02287. If we have a more potent compound, maybe we can come up with a more convenient dosage formulation, or maybe its potency leads to another indication, as we might be seeing in one particular case in our portfolio. So when we talk to partners about GT02287, it becomes a broader conversation about, well, we have GT02287, but it's kind of a package deal. And part of that package is GT02287 is not only applicable for Parkinson's disease, but in other neurodegenerative diseases where we see GCase playing a prominent role. Alzheimer's disease is one. ALS is another. Gaucher's is also something that we would probably take a look at as well. And we need partners who appreciate the promise and potential of GT02287 broadly by focusing it first on Parkinson's disease, but not limiting what we're willing to by not limiting what the potential value of the entire portfolio is to us and our investors. Does that make sense?
Gene Mack, CEO of Gain Therapeutics. Gene, thanks so much for your time today.
Appreciate that. Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.